Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to another episode of the Genetics Podcast. I'm really excited to be here today with Dr. Jeffrey Chamberlain, who's professor at the University of Washington School of Medicine, where he's the head of the Muscular Dystrophy Research Center. He's also director of the Wellstone Muscular Dystrophy Center. Dr. Chamberlain is one of the true pioneers in gene therapy for muscular dystrophy. We're going to go back initially here to the late 80s, early 90s, where he was one of the pioneers in early multiplex sequencing of the gene and also laying the foundation for some of the gene therapy innovations that we're going to talk about today. So Dr. Chamberlain, thank you so much for joining us, taking the time today.
Thanks. It's great to be here.
As I promised in the intro, I wanted to go back to the late 80s, early 90s. I'd love if you could just paint a picture of, at that time, what were the big challenges that you and your colleagues worked on? And maybe you could, if you can, share when you learned, what we learned about the structure of the gene, how it was related to disease and some of the opportunities for therapeutics. What did you imagine then that has come to fruition today? And then what are some of the things that are either still stubbornly out of reach or they just haven't happened yet because we don't have the right technology or understanding? So maybe you can just take us back to that time and then bring us back to the present.
Sure. Well, it's been a long journey since those early days. The late 1980s were a really exciting time in the genetics field, although I think they've remained pretty exciting since then. But what was particularly exciting from my point of view was that the gene that is defective in Duchenne muscular dystrophy was isolated. And that was work that was done by Lou Kunkel's lab at Harvard Medical School and almost simultaneously by Ron Wharton at the Hospital for Sick Children in Toronto. And what was really exciting about that was a couple of things. One was that Duchenne muscular dystrophy is one of the most common inherited disorders. It's a lethal genetic disorder that affects children. So there was, it was getting a lot of attention at the time, the disease. And, it was in the early days of genetics where very few human disease genes had been isolated before. And nobody really knew that much about the Duchenne muscular dystrophy in terms of the cause. What was the gene? Nobody knew what the protein was or anything like that. And the way that the gene ended up being cloned was through, I think, what came to be known as positional cloning, trying to clone a gene based on its chromosomal location and linkage analysis and things like that. And so Lou Kunkel published the first data suggesting he had the gene in about 1986. And, in my mind, that was really kind of the beginning of the Human Genome Project, because that set in motion this idea that you could isolate genes for different genetic diseases, you could start characterizing them and things like that. But, at the time, there were still fairly basic tools in how to go about analyzing the gene. And it turned out to be a particularly difficult locus to study because what we now know is the dystrophin gene, as it came to be known, is the largest gene that's ever been found in nature. It's more than 2.2 million base pairs in size. Wow. So as you can imagine, it took quite a long time back then to work out the structure of the gene. So, I had actually begun my postdoc about the time that the Kunkel cloned the gene, and we were in touch with his lab. And, I've got to say he was incredibly generous with his tools and his clones. And he immediately sent us some fragments of the cDNA because at the time, that's all they had. They only had partial cDNA clones. And we used that to do two things. One, we began trying to map out where this same gene was in mice in order to try to determine if this particular mouse model known as the MDX mouse that had been out for a couple of years might actually be a mouse model for Duchenne muscular dystrophy. We were able to show that, in fact, the MDX mouse was a mouse model for Duchenne muscular dystrophy, and we started cloning out the mouse cDNA. And that work actually was important for my future career after I left my postdoc, because we took advantage of the mouse models and the mouse cDNAs to start understanding the structure and function of the gene, what it does, and that led into my interest in gene therapy. In the meantime, though, I was at Baylor College of Medicine, and they had a big DNA diagnostics lab there at the time. And at the time when you isolated a new disease gene like this, there wasn't a whole lot you could do in terms of therapies or things like that. So it seemed like the most immediate impact of isolating this gene was to develop DNA diagnostics, because until then, diagnosis of Duchenne muscular dystrophy was quite difficult. It's an X-linked recessive disorder, and 1/3 of all cases arise from a spontaneous new mutation. So the consequence of that was that fully 1/3 of all the cases arise in a family with no history of the disease. And typically, the kids that have the disease are not even picked up until they're 3, 4, sometimes 5 or 6 years of age. And a real problem with that, not only for the family, but by that time, sometimes these families have had additional kids. And, I've met some families that have, 3 different boys with Duchenne muscular dystrophy. So we were really interested in developing DNA diagnostics to be able to do prenatal diagnosis, but also carrier detection.
Right.
And those are a little more applicable if there's a family history. Someone in the family has Duchenne muscular dystrophy, so then being X-linked recessive, all the female relatives are potential carriers. But there wasn't a way to figure out who was a carrier, who wasn't, and all that. So we simultaneously, with our work on the mouse gene, started cloning out parts of the human gene. And again, it was a bit of a challenge because it was a 2 million base pair gene, and even by 1990, only portions of the gene had been isolated at that time. But what was coming out from early work with Southern blot analysis, which was the state of the art at the time, was that 2/3 of all the patients had deletions that took out portions of the gene. So we started going through and trying to clone out the portions of the gene that map to the more frequently deleted regions. And we reasoned that, it was just very cumbersome to be doing all these Southern blots throughout what was turning into an enormous gene in order to look for deletions and also some duplications and things like that. So around that same time, Kerry Mullis had just discovered the polymerase chain reaction. And the guy I was working for with my postdoc, Tom Caskey, was actually on the scientific advisory board of Kerry Mullis's company, which at the time was Cetus. So he knew about PCR from the beginning, and he brought Kerry Mullis over to our lab, and we talked to him a lot about how could we potentially apply PCR to Duchenne muscular dystrophy diagnosis. And so and then we started cloning out different regions of the gene. So we were setting up PCR reactions for these different regions. And even that was very cumbersome. So we got the idea, why not combine some of these PCRs? And at that time, nobody had ever PCR amplified more than a single isolated piece of DNA at a time. So we started combining these, and we developed a technique that we named multiplex PCR, which probably seems pretty obvious today, as many of the things we did back then. My graduate students, I talk to them today, and they go, yeah, really? That took you more than a week to do that? But anyway, we eventually got it going where we could simultaneously amplify 9 different segments of this gene. So we were really just scanning small segments of the gene, but they were all picking up these different deletion hotspots. And we got to the point where we could pick up more than half of all cases of Duchenne muscular dystrophy in a 3-hour PCR reaction. And so we began applying that to, again, prenatal diagnosis and carrier detection. And that really set things in motion. And the multiplex PCR method then started being applied to a lot of other genes. And
Right.
in terms of Duchenne diagnostics, I think it was the most commonly used method for about 20 years until much more advanced and easier to use methods came along after that. And so it's kind of obsolete now, but it was fun in the early days working with that, and I think it was useful. Yeah,
good 20 years. And so I
Yeah.
guess it was very clear that deletions were concentrated in specific exons like 45, 51, 53, so you could design the assay around those known hotspots, right?
Exactly. Yeah. And so that's what we did. If we'd had to amplify, all what we now know are 79 exons, I probably would have given up.
Yeah.
But,
Well, I mean, what did you know at the time? Because most of the people listening to this podcast won't have been in the field at that time. Did you have any concept of how many exons there might be? Or I'm just really interested to understand how you made sense of this with the tools that we had at the time.
Yeah, we did not know how many exons there were. What we knew was that the gene was, more than several hundred kb in size. Around the time we were, had already started doing some of the PCR. Lou Kunkel's lab put together and ended up cloning out the entire cDNA. So at that time, we knew, that the cDNA was 14 kb, but we still didn't really have a good feel for how large the gene was. But you could go through, and we fortunately at Baylor College of Medicine, we had access to hundreds of DNA samples from different patients and carriers. And by going through and initially doing Southern blots, we could see that there were deletions in some patients, not in others. Some patients were deleted for the amino terminal part of the cDNA. Some were deleted for the C terminal part of the cDNA, and they were just scattered all over the place. So what we were able to do was to zero in on, as you mentioned, there were two major hotspots of deletions where, 2/3 of all the deletions mapped right into the region that we now know is encoded by exons, I think 44 to 51. And then another third of all the deletions were encoded in the region around exons 3 to 10. So not knowing what those exons were, how many there were, we just took the cDNA that corresponded to those regions and screened lambda genomic libraries, phage lambda libraries, isolated the clones and went in and using hybridization with the cDNA, we're able to find the exons. And then we sequenced each exon in the flanking regions and used that to develop the PCR primers. So we were kind of working blind. We didn't know exactly what part of the gene we were in, but we knew that it mapped to the hotspots.
Yeah, it's
And it
amazing.
was it was a labor intensive work because as you can imagine, you pull out a phage lambda clone that is maybe 18 kb in size, and generally every clone we isolated only had one exon on it because the gene is so large because the introns are enormous. So you got this 18 kb clone and there's maybe 150 base pair exon in there, and you got to kind of isolate it, subclone it, and sequence it. And, back then we were doing manual Sanger sequencing and with these giant gels and X-ray films and all that kind of stuff. So it was interesting work. So.
When did you start thinking about therapeutics and how early did that come in, and what was the spark that had you thinking about micro-dystrophin, exon skipping, potentially some of these other approaches that we now see today?
Well, we kind of had the idea of gene therapy in the back of our mind really as soon as the gene was isolated. The issue is we had absolutely no idea how to go about doing gene therapy.
Right.
But, that was also the early days of gene therapy. My postdoc mentor, Tom Caskey, was one of the early pioneers in gene therapy. He had isolated the HPRT gene, which is defective in Lesch-Nyhan syndrome. And, I think that was the first human disease gene that was manually cloned. And so Tom was always talking about doing gene therapy for Lesch-Nyhan syndrome. So it was kind of something that people in the lab talked about, but at the time I wasn't sure how realistic it was. And so, I thought the most productive thing I could do would be to try to characterize the mouse models and use that as a system to understand what does this protein actually do and why is it when it's missing you have to get this terrible disease. And I kind of thought, let's just try to understand the protein, develop a little better vision of what does it do, what tissues is the protein produced in, and what's going wrong. And but we thought eventually maybe if we learn this and we figure out where the gene needs to be, and maybe someday we can figure out a way to deliver genes to muscle, perhaps gene therapy would come online. And we actually wrote a review article in 1989 that kind of talked about what was known about Duchenne and the dystrophin gene at the time. And at the end of that article, I kind of went out on a limb and I said, this is a genetic disease. It's recessive. Mutations don't produce a protein. They don't cause any toxicity. This would be an ideal candidate for gene therapy. Of course, we didn't know how to do that. We speculated a little bit, and pretty much everything I speculated on would never work. But, it sort of planted the seed in my mind. And then I went and set up my own laboratory. I got a faculty position at the University of Michigan. And my original plan was really just to do basic mouse genetics. And so one of the things we started doing was we actually were able to put together the first full-length cDNAs. The Kunkel lab had only cloned out a bunch of fragments of the cDNA. We were able to assemble them into full-length clones. And then we started making transgenic animals. And the idea was, well, we have this mouse model that doesn't make the protein. Let's put the full-length cDNA back in because we still didn't have the whole gene. It was 2 million base pairs. So, let's just make transgenic mice and ask what are the most important parts of the protein. And we knew that not all of the parts of the protein were really important because the early deletion work by Lou Kunkel and by Kay Davies at Oxford and others had shown that a number of these patients that had intragenic deletions actually had very mild versions of the disease. Most of them had the full-blown Duchenne muscular dystrophy, but there were these rare patients that had deletions that took out part of the gene and part of the protein coding region that still had mild versions of the disease. And that suggested that this was a pretty flexible protein. And on the one hand, from a gene therapy point of view, it suggested maybe you don't have to deliver the entire gene to have a therapy. But also, we were just interested in, well, why is this protein so flexible? What are the really important parts and what do they do? Do they interact with other proteins? We didn't really know anything about it at the time. So we started making transgenic animals. We started making deletions in our cDNAs to somewhat correspond to some of the deletions in the mildly affected patients. And we, laboriously generated transgenic mice, back-crossed them, did all that stuff, and worked up the phenotype and asked, well, which animals have muscular dystrophy? Which ones have some of the symptoms, but not all of them? Things of that nature. And the other thing we also started asking, well, what tissues do you really need to make this protein and to impact the disease? We didn't know how much of it was pure muscle, how much it was neurological, were motor neurons involved, Schwann cells, things like that. And none of that had been worked out. So one of the first things we did was we said, well, let's make a transgenic mouse that only makes dystrophin in muscle tissue. And we had a convenient way to do that because even before I knew anything about Duchenne muscular dystrophy, I had worked in a muscle developmental biology lab, actually with Steve Hauschka, who's here at the University of Washington still. And my thesis project was I cloned the gene for muscle creatine kinase. And at the time, that was only the third muscle gene that had ever been cloned. And we used that to identify the promoter and the enhancer elements of the MCK gene. And that was the identification of the very first muscle enhancer element. Tells you how long ago
I worked on enhancers in my PhD, so I should know that history, but now I do.
Well, yeah. Once again, I tell this to people in my lab and they go, yeah, what'd you do the next day? So. But anyway, so we went back and started collaborating again with my graduate mentor, and we used these MCK promoter enhancer elements to drive expression of our cDNAs in the MDX mouse, the mouse model for Duchenne muscular dystrophy. And we started learning a lot about the protein. But the first thing we learned was that we showed that if we expressed the full-length protein only in striated muscle, we completely eliminated all features of the disease, at least all the ones that we could assay at that time. So, on the one hand, I started thinking, oh, we just cured this mouse. We're doing gene therapy. And it actually made a pretty big splash in the field. I don't know if you remember back far enough, but at the time, the big supporter of muscular dystrophy research was the Muscular Dystrophy Association. And it was headed by, the comedian Jerry Lewis, who was a former movie star, or I guess he still was a movie star, but Jerry raised several $1 billion to support research into muscular dystrophy, and he put on this annual telethon that was broadcast all over the world. And so they got really excited, and they said, this work that you just did is the first demonstration that muscular dystrophy can be treated. Nothing like that had ever happened before. And it was kind of surprised me because at the time, I didn't think it was all that big of a deal, but whatever. And then Jerry Lewis invited me to appear on his telethon and kind of totally freaked me out because I'd never been on TV before. And before I went up on stage, he turned to me and he goes, don't be nervous. There's only 70 million people watching, So that was fun. And so we went back and did that a couple of times. And it gave me an opportunity to really try to explain the science we were doing and the emerging gene therapy field in fairly simple terms to a pretty large and completely lay audience, people that didn't know anything about the science and many of which really didn't care that much about the science. So they'd always tell me, try to keep it under a minute, that's otherwise you're going to put everyone to sleep and all that. But anyway, at that point, we realized we're kind of doing gene therapy, so let's just stick with it. And, but we didn't know how to deliver genes to muscle. There vague ideas. You could take a syringe and jab muscles all over the body a few thousand times and maybe get a gene in there. But, really taking that to the next level and having something that would really work, in the early 90s, we had no idea how to do that. And, one of the things I tell the folks in my lab, the students and the postdocs is, if I sat down and thought really hard about how can we really do gene therapy it was just overwhelming and I would have given up. And so I said, the key to this is not to think too far ahead, really
Yes.
just set your goals in manageable units and figure out, what can we do over the next couple of years? And then we'll see where we're at. And then we can figure out what to do after that. And so, it just started an incremental process to try to understand the gene and use that information to eventually get towards gene therapy.
Yeah, I mean, what is amazing to me about this story is because I think if my understanding of the timeline is right, AAV really they didn't start to solve the manufacturing scale challenges and really do first therapeutics even in animal models until the mid 90s to early 2000s, right? And so we'll get onto this, but micro-dystrophin becomes really important because you can't fit this whole gene into an AAV vector, but you're doing this work really to understand the fundamental biology of the gene by knocking out different exons and figuring out what works where. But it seems like that turns out to be a really critical piece of insight that paves the way for the next steps. Were you aware that a microdystrophin or some similar concept might be therapeutically useful, or was this one of those moments where two interesting ideas come together and you figure it out?
It was really two things that sort of came together independently. As I mentioned, part of the focus of my lab, I mean, I'm a PhD scientist, so I figured, I got to continue to do basic science. So, we were somewhat grounded in really structure function studies of dystrophin, really trying to identify what do the different domains of dystrophin do, what are the most important ones, and things like that. We were simultaneously trying to develop methods for gene therapy. But, initially we thought we really needed to deliver the full-length protein or at least a half-size protein, which, as you know, would still be too big for AAV. But at that time, in the mid-90s, hardly anyone was even working on AAV. It wasn't on our radar yet. The systems that were attracting a lot of attention the initial one was just plasmid injection and then plasmid electroporation. John Wolf's lab at Wisconsin had shown that plasmid DNA, when injected into muscle, was surprisingly stable, and you could get stable expression for over a year. And that led to actually a human clinical trial in Duchenne at Genethon in France, where they were just doing intramuscular injection of plasmids. As you can imagine, it didn't go that well, but nobody was harmed in the process, I guess.
Yeah.
But then people became interested in retroviral vectors. The hematopoietic field was doing a lot of stuff with retroviruses, as you know, for SCID and other diseases. But, retroviruses, again, injected into muscle just wasn't very efficient. You did not get widespread delivery. We then moved on to adenoviral vectors. That was the big rage in the mid-90s. Adenovirus actually gets into muscle quite well, but again, only by intramuscular injection. And a lot of the early adenoviral vectors were immunogenic. You'd get beautiful expression for a month or two, and then it was gone. So we started, like a lot of other people, started trying to modify adenovirus, removing as many of the viral genes as possible to reduce immune responses. We developed what we called a gutted adenoviral system. Other people did the same thing. Sometimes they're called helper-dependent adenoviruses. That helped a lot. With the adenoviral system, we could actually fit full-length dystrophin in there, and we could get expression for over a year, but it started fading away, and we're still getting some immune responses, so it was just not an ideal system. And at that same time, the basic research part of my lab developed these microdystrophins, and part of it was that we got to the point where we wanted to ask, you can delete half the gene and it still works really well. How small can you go? What is the minimum size of a dystrophin that would have any effect at all? And that led to the development of microdystrophins that were just under 4 kb. And we actually made even smaller ones, but they didn't work. So what we eventually started calling microdystrophin, which was, we named it micro to distinguish it from the mini dystrophins that were in the patients. The smallest one we could get to work was, again, a little under 4 kb. And it was around that time when, Barry Byrne and Jude Samulski and Jim Wilson almost simultaneously each published a paper showing that AAV vectors, when they were injected in the muscle, did not elicit an immune response and were stably expressed for over a year. So we got excited about that and realized our microdystrophins would fit into AAV and started doing some testing there and intramuscular injection, and showed we could actually make microdystrophin in the muscles. But again, it was intramuscular injection. It was not spreading. And at the time, a lot of people were going, no problem. It just maybe 3,000 injections into each kid who got this cured.
Oh my
And I was thinking,
gosh.
this is never going to work. And not only that, but you'd never be able to hit some of the critical muscle groups like the heart or the
Yeah.
breathing muscles, the diaphragm, the intercostal muscles. So we figured, okay, AAV looks pretty good. Micro-dystrophin looks pretty good. But we got to find a way to do systemic delivery and really get it body wide. And so we turned our attention to trying to do intravascular delivery. And at the time, everything we infused into the mouse went to the liver. We just couldn't get anything to muscle. And part of the limitation was, the production capabilities of AAV at that time, this is in the late 90s, were still fairly primitive and all that. But labs started getting better and better at making high titer, high quality preps of AAV. And we were fortunate there were a couple of good AAV groups at the University of Washington, where I had recently moved, particularly David Russell, who was a big AAV guy. He trained with Dusty Miller at the Fred Hutchinson Cancer Research Center. And David actually cloned several of the early serotypes of AAV, and he isolated AAV6, which he gave us and turned out to be a really good vector for muscle, had a lot of tropism for muscle. And with David's help, we figured out ways, to increase our titers of our vectors by over 100-fold over what we had been able to do. And at that point, when we went back in and did intravascular delivery, suddenly the vector was going to muscles all over the body. So we realized there was this threshold effect where if you had to get to a certain critical dose of the vector within the vasculature, then it started leaking out of the capillaries and infecting the underlying muscle tissue. And so in 2004, we published the first paper showing that you could deliver genes body-wide to muscle using AAV. And I think that was a real breakthrough because we also showed we could deliver the microdystrophin using these muscle-specific promoters, and it was all came together so it fit into AAV. And, now there's, more than 10,000 kids have been treated for several neuromuscular diseases using this systemic delivery of AAV method that we published back then, and it's it's shown a lot of promise. It's not perfect, and in fact there are some issues in the clinic that need to be solved, and a lot of our work is focused on that at the moment.
This is probably a really good segue to the present. You of all people, I want to really have your perspective. Right now, we have, I think the field of gene therapy in general is in a pretty critical moment. You've got some amazing progress. I think you shared an example there. We've got more than 10,000 people with neuromuscular diseases that have been treated. There's also safety challenges, funding challenges. This field goes through cycles historically. And I'd just love to have your perspective on what's really working today, and people need to see through the short-term challenges, and there's going to be continued opportunity to help patients here. And then on the flip side, what are the big challenges that we still really do need to reckon with and overcome?
Right. Well, just to focus on the successes, first of all, I think really the most dramatic success possibly in the entire gene therapy field is in an area that I wasn't working on, but is spinal muscular atrophy. And, one of the best treatments now for SMA is a gene therapy drug called Zolgensma. And they try to treat these kids as soon after birth as they can because it's a very rapidly degenerating disorder. But again, they're doing a intravascular delivery of AAV9, but delivering the SMA protein. And I think I've lost count, well more than 5,000 kids have been treated with that drug, and it has a major impact on their lives. I mean, it keeps them from dying by age 1 or 2. It's not a complete cure, but it's pretty darn good. So that's really the poster child so far, and it's something interesting maybe about treating essentially newborns with this drug, with these AAV vectors, these high doses. It seems to be fairly well tolerated in the neonates and all that.
Do you have any idea why that is, or why that might be?
I'm not sure.
Me neither.
If they have a less developed immune system, perhaps, I'm not really sure. In contrast, though, the issue with Duchenne muscular dystrophy now is at last count, there's, I think, almost 1,500 Duchenne boys have been treated with AAV microdystrophin. And it's interesting. There's actually, as far as I know, there are 7 different companies that are in the clinic doing AAV microdystrophin, and each one has a slight, variation on the theme, either a different serotype of AAV or a different promoter or a different type of microdystrophin. There's a lot of ways you can make microdystrophins and all that. So, there's a lot of different competing versions of it. Nonetheless, I would say the impact in the clinic has not been as impressive as what we've seen in the animal models. Certainly, certainly in the mice, we can go in to mice and give a high dose and essentially cure the mouse of the disease. There's been studies in a rat model of Duchenne. There's a couple of dog models of Duchenne. You have a tremendous impact on the disorder in all those models. But in the clinic, it's been, reasonable, but not nearly as dramatic as what you see in the clinic. And I think part of the reason for that, well, there's two reasons I would say, is that the amount of microdystrophin that's being made in the muscles of the patients is significantly lower than what we get in the animal models. And we're not entirely sure why that is. It may just be that the dose you need in humans is higher than it is in all the animal models that have been tested. Every species is a little different. The other thing is, maybe also relates to the age of the patients. All the Duchenne trials have typically started with boys that are 4 years of age or older. And I think it's becoming clear that maybe it's a little harder to get the vector into boys at 4 years age, maybe compared to the newborns with spinal muscular atrophy. The other thing is there's already a reasonable amount of dystrophic degeneration, even in the 4-year-old boys, and there's a significant inflammatory environment with a lot of immune cells, fibrosis setting in. And I think all of these are creating an environment that's a little less susceptible to, one, getting the vector in there, and then two, actually being able to reverse the disorder. So those are some challenges. In the best case, some of these boys are stabilized. The progression of the disease is significantly slowed down. Some show a little bit of an increase in strength. Many of them do not, though. And one of the things I think that's coming out is that there's also a lot of variability from one kid to another in terms of how much vector they take up, how much of the dystrophin protein they're making. Now, some people have also argued, well, maybe microdystrophin's too small. It just doesn't work in human muscle the way it works in dog muscle or mouse muscle. I think that's partly true, and we're trying to come up with better ways to deliver more potent dystrophins. But I would argue the major problem, I think, is the delivery efficiency. And, if we can deliver more microdystrophin, I think it's going to work a lot better. We've got two companies out there now, Kinea Bio and Solid Biosciences, that have come up with better ways of formulating their AAV and delivering it, and they're now seeing significantly higher levels of dystrophin. We're not seeing any functional data yet, so it's going to be interesting.
Yeah.
Does it really work better now that they can make more, or is there still a problem? We'll see. It's a work in progress. The other thing is, you could argue, well, if you're not making enough dystrophin, why don't you increase the dose? And that gets into perhaps the most important problem at all, is these doses are already enormous. The initial doses that we published on in mice was 4 times 10 to the 14th vector genomes per kilogram body weight. The early clinical trials used half of that, 2 times 10 to the 12th, and a lot of them are now down at 1 times 10 to the 14th. So the doses come down for safety reasons. And the problem is if you increase that dose, you would get a lot more dystrophin in it, but you can't because of the adverse events that are appearing in the clinic. And we're seeing, some liver problems and some vascular problems, thrombocytopenia, some complement activation, innate immune responses, not in most of the patients, but in a subset of the patients. And Sarepta Therapeutics, unfortunately, had three of their patients died earlier this year in their clinical studies. So, and I think it's coming clear with some other gene therapies for other diseases that when you need these high doses for systemic delivery up above the 10 to the 14th vector genomes per kilogram range, you are right on a knife's edge where, most of the patients are going to do okay, but some of them are going to have very severe adverse events and some of them will die. So that's obviously not acceptable. So we need to find ways to lower the dose is the number one challenge. Secondly, we'd like to be able to deliver more potent forms of dystrophin. And third, I think people should start looking into, can we treat younger patients and maybe be able to prevent the disease from coming on rather than reversing it. So those are the kind of things that occupy my lab and many others in the field at the moment.
What are your thoughts on gene editing rather than gene therapy? And where the question is coming from is, do we have, it sounds like there's some, there's some science and engineering problems in gene therapy that actually, if we can solve some of these safety issues, improve the amount of dystrophin, micro-dystrophin that's getting in, then there's a
Right.
really viable path there. But then there's also always new technological leaps, gene editing maybe being the most
Right.
obvious one, but there could be others that you're thinking of as well. So curious to get your thoughts on those.
A couple of things. I mean, I think gene editing has tremendous potential. Obviously, it's revolutionized therapeutics and basic biology, and at the same time. So, if we could get in there, the promise is that rather than doing gene replacement, you're actually doing gene repair, hopefully, or modifying the gene to allow dystrophin to be made. You know, I think it's going to have a huge impact on the field. I would say it hasn't yet, and it's going to be a while. We used to get a lot of criticism in the 90s on how gene therapy was like the most overhyped and overpromised thing on the planet. Well, I think we can now say that gene editing has surpassed
Taking the
those
crown.
lofty goals. Some of the exaggeration I hear about the potential is mind-boggling, and so, some of the false promises that are made to patients is really sad, and people need to be very careful about that. But in any case, it has a lot of potential. So, there are challenges. Some diseases are obviously easier to treat with gene editing than others. There's a lot of work going on the Duchenne gene with gene editing by many labs, and it's coming along. It is getting there. But one of the big challenges you face is, again, it's a 2 million base pair gene. It's a new mutation syndrome. There are mutations all over the gene. Many of them are small. A third of the patients have point mutations. Two-thirds have deletions or duplications within the gene that can be anywhere along this 2 million base pair locus. So the early applications of gene editing were primarily using CRISPR-Cas9 to try to go in and mutate an exonic splice site, with the idea, sort of like doing exon skipping. If, let's say, you had a stop codon in exon 50, you could go in and mutate the splice acceptor site using Cas9 and allow then the pre-messenger RNA coming off that edited gene would skip over exon 50, and you would make a slightly smaller protein, but an almost full-length protein. And so, this was raised the idea that, hey, we can get beyond micro-dystrophin, we can make full-length dystrophin and all that. But, that's only applicable to a subset of the cases, and the kids that have these large deletions are not going to make full-length dystrophin. And the other thing is, it was estimated that you would need to come up with 20 different CRISPR-Cas9 vectors in order to treat something like 70% of the patients. And, that means different guide RNAs, things like this. So you're looking at companies having to develop 20 different gene therapy vectors, get them all tested and FDA approved. And boy, that's a big challenge.
Tall order. Yeah.
So typically what the companies are doing is, it's sort of like the exon skipping field. We didn't really get into exon skipping, but there's another form of therapy out there is to use antisense oligos and just inject these into the kids, and with the idea they'll get into muscle, they'll do the same thing, bind to splice sites, skip mutant exons and all that. But typically it's, everybody's focused on the single most deleted exon in the gene, which is exon 51. And they say, well, if we can get this treatment working, we can treat 15% of all the kids. Well, that's great, but there's still 85% that you're not treating and all that. And there's now exon skipping drugs out there for, I think, 4 different exons. Similar to the microdystrophin, I don't think they work. I haven't seen any good evidence that they make significant amounts of dystrophin or have a significant clinical impact. And, Sarepta Therapeutics, that owns most of those, had a press release a couple of weeks ago where they said we're not getting any statistically significant data suggesting we're having an impact on the patients. Hasn't caused them to pull any of these drugs off the market, but we'll see what happens with that. And so gene editing, though, potentially can be more efficient. The problem with exon skipping so far is these antisense oligos just don't get into muscle very well. And again, they get into mouse muscle pretty well, but they don't get into the patient muscles that well. So gene editing is now typically applied with AAV vectors. So rather than delivering a microdystrophin, you're delivering Cas9 and a guide RNA. And the efficiency is enormous. You can really get it in there, but you still got to get up to the 10 to the 14th vector genomes to get Cas9 into muscles all over the body. The other problem we're facing is that The advantage of AAV is you get stable expression, right? So, we've seen some of these kids in these Duchenne trials are still making pretty reasonable levels of dystrophin 5 years after the infusion. There's studies in dogs showing stable expression for 10 years of micro-dystrophin. But, do you want to have Cas9 floating around every muscle cell in your body for 10 years and editing whatever it jumps, it hits? So that's a problem. The other problem is that, these Cas enzymes are all bacterial enzymes. They're immunogenic. So you really got multiple strikes against you trying to apply systemic gene editing for Duchenne muscular dystrophy. Now, there are ways around that, and some of the things are coming together for both gene replacement and gene editing. In that, one, there's a whole new class of AAV vectors that have come out over the last 2 or 3 years called myotropic vectors that seem to be able to get into muscle at doses at least tenfold lower than the ones that are currently in the clinic. And that gets you down potentially to a much safer level. Two, a lot of people are now working on ways to try to get inducible expression of Cas enzymes or to be able to shut it down, because it looks like you really only need transient expression. If you can make your Cas9 or whatever Cas you want to use for 2-3 weeks, that's probably all you need. But then you want it to go away and not stick around and edit other genes or cause an immune response. And then there's better Cas enzymes. Cas9, I think, is not going to work. It's a double-stranded nuclease, and there's increasing studies showing that this can lead to chromosomal rearrangements. Everybody used to focus on off-target base changes, but now what we're seeing is inversions, deletions, translocations to the point where I'd be surprised if the FDA ever approves systemic delivery of Cas9, although I'm surprised sometimes what gets approved and what doesn't. But the field is shifting now. I think, there are better methods like base editing is significantly safer. It doesn't induce double-stranded breaks. It does induce single-stranded breaks. Some of the work coming out from the labs I've seen working on base editors is they're actually more efficient than Cas9 at inducing base changes. It opens up more targets, I think, than the double-stranded breaks. And again, it's more efficient for whatever reason. Prime editing may be even better, but prime editing is a real challenge. It's hard to get to work in vivo. It's a pretty complicated thing, but it's coming along. It's getting there and all that. So, eventually these things I think are going to allow for some clinical trials with gene editing. Now, there is one company in China that reported at the gene therapy meeting last year in New Orleans, the American Society for Gene and Cell Therapy, that they had attempted a gene editing human clinical trial. They treated 2 boys in China with a Cas9 treatment to try to target exon 51. To be honest with you, I was appalled that they would do that. I don't think it's safe. I don't think it's ready for prime time. And, like a lot of companies, they got up there and said, look, it's working. We got dystrophin. And, I'm looking at their slides. I go, I don't see any dystrophin. I don't know what you're talking about here. Mm-hmm. And I met with them afterwards and they're going, oh yeah, we're talking to the FDA. We're going to scale this up and treat people right and left and all that. And I go, please don't, you don't know what you're doing. So we'll see what happens with that. I am worried about it, but we need safer vectors and all that. The other thing is eventually for gene editing, what we need is more of a universal system to treat many patients at once. And there's a couple of labs now working on methods to try to, rather than fix individual mutations, to actually insert cDNAs to replace large segments of the gene so that you could potentially have, 2/3 of all the patients out there with a single vector that would come in and just swap out any region of the gene with a larger cDNA.
Who's working on that
way? Well, there's a couple of labs working on that. The main one I know of is Courtney Young down in San Diego. She's got a company down there, Myogene, that's working on that approach. There's a couple of other people, I forget their names, but and other groups are trying to develop this for other diseases too. The idea of either to use a HDR recombination or HITI methods to cut and paste things into the gene. Well, we'll see how well they work. The efficiency is a little low, but ultimately that may be the way to go.
Yeah, amazing.
The other method I'll say briefly is what we published on a couple of nice papers recently we figured out ways to deliver larger dystrophins now. So we think we can go beyond micro-dystrophin, but the challenge is that we have to use multiple AAV vectors. And there's a couple of ways to do that. There's been published methods using homologous recombination or trans-splicing or things like this. The method we've latched onto more recently uses split inteins. Mm-hmm. And the way split inteins work is you can produce 2 different proteins, and if they each have a split intine on one side, those half proteins will come together inside the muscle cell and ligate together and make a larger protein. And with 2 vectors, we can make but the mini dystrophins that are double the size of the micro-dystrophin. And with 3 vectors, we've shown we can make large amounts of full-length dystrophin and body-wide and in the mouse models. So we'll see now whether we can scale that up and get it into the clinic, but we're very excited about that at the moment. Yeah.
That's amazing. And you actually just perfectly anticipated my last question, which was going to be about what kinds of things you're focused on and what's exciting for the future. Is there anything else that you want to leave the audience with before we close out?
Well, I would say that, so far, gene therapy has not been the miracle cure for muscular dystrophy that we hoped it would be, but we're getting there. We're making progress. We've learned a lot about how to do the clinical trials, how to deliver AAV to the patients. The FDA is starting to come along and accept some of these approaches. But perhaps most importantly, we're developing a much deeper understanding of the limitations. And those relate to, I think, high doses, limited transduction, and maybe less potent dystrophin cassettes than we need. And ways to solve those problems are falling into a place. There's still a lot of labs working on it, making great progress. And I'm optimistic for the future. We're not there yet, but we're getting there. So. Yeah.
Amazing. Well, Jeff, thank you so much for sharing all the time and insights with us today. This was an amazing conversation.
Yeah, thanks. Enjoyed talking with you. Yeah.
And thanks everyone, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.